here we are. The third group of macromolecules that we are going to talk about today. And these fit into the true monomer-polymer motif. And we'll talk about a lot of chemistry that holds the proteins together and a little bit about what they do. And we'll be hearing about proteins all year long. They're very important in biology. So here's the basic building block of a protein. This is called an amino acid. It has a constant in that this amino group, you'll recognize from our functional group work, the nitrogen is connected to a central carbon. That central carbon makes 4 bonds. One is a hydrogen all the time. The other one is always to the carboxyl group, the C double bond O OH. Depending on the pH of the system, you can gain or lose that hydrogen, and that's the acid. That's the amino acid side, amino part acid side. There are 20 different R groups. R is just like X and whatever you want to call it. This is the variable. 20 different ones that we care about in the proteins that we're going to talk about in our scope here. We'll get into them in a second. So first, before we talk about the building blocks, let's talk about all the different kinds of roles proteins can serve in an organism. They're varied. We're just touching on them. We're just trying to give a few examples here to kind of put it in perspective. We'll very soon be doing a whole chapter about enzymes. Enzymes are proteins, basically. They're this huge molecule that can help catalyze reactions. You remember that there are catalysts in chemistry. The biological catalysts are enzymes. They're very specific. We'll get into a whole lot of details about those guys soon. Proteins can serve as defensive proteins. We'll talk about antibodies. There are plenty of other ones too, but antibodies we'll talk about when we get into immunology. They're the thing that are circulating in your bloodstream and help protect you against viruses, bacteria, and other pathogens that might sneak in. Proteins can serve as storage proteins. When you cook an egg, the white part of an egg, when you cook it, is albumin. And it solidifies when you cook it. We're going to do an experiment to take a look at protein structure, and we'll use the egg white actually as our source of protein. It's a source of energy for the body. We break it down and we can use it for energy, but it's mostly used as the raw material to build up new proteins. If you eat proteins, you chew them up in your stomach with enzymes, you chew them up with your small intestine further with enzymes, and then you reuse the pieces, these amino acids. You'll see in the chapter, I think it's chapter 5, we'll talk about transport proteins or transmembrane proteins, different proteins that are embedded in the cell membrane. They do all types of work. Sometimes they also act as receptors as well. Oh, there it is. They work as receptors as well. So proteins serve lots of roles on the surface of a membrane. We'll get into that very soon. Later on, we'll talk about hormonal proteins. Hormones can be steroids. You've already met estrogen and testosterone. They're sterols in the lipid family. There are also protein hormones. Human growth factor is a protein hormone. Insulin is a protein hormone. We'll get into insulin and its role in sugar regulation later on in the year. Our muscles move because of the action of 2 very important proteins. Actin, up here, these little balls, and myosin, this purple protein. Again, we'll use examples of how these guys work later on, but know that your muscles move because these 2 proteins slide against each other and contract. And then finally, structural proteins. Our body is made up of all kinds of things. Bones are the things you always think of for structure, but we also have sort of softer structural protein. Collagen is the stuff that makes you nice and squishy. Your ears, I can't touch them with my headphones on. And then also keratin is in your hair. Your hair is all made out of proteins, and I'll talk about that again in a little while. So back to our amino acids. So we have these 20 different building blocks. Think of them as an alphabet, so to speak. They all have different flavors. I don't want you to memorize the 20 amino acids, but what I do need you to memorize is their flavors, so to speak, or their chemical properties. There's a large group that are hydrophobic. We'll get into that in a second. A large group that are polar, and then a smaller group that are actually charged. So first let's look at the hydrophobic amino acids. So remember, we have this constant area, and I love how the book color codes things to remind you. This is the backbone, the NCC. So the amino side chain, amino group, the central carbon, the carboxy end, and then the R group. The very simplest amino acid is called glycine. It has just a hydrogen there. Hydrogen and carbon, remember their electronegativities are very similar. So this is these are considered a non-polar because they are not polar. They don't have a polar bond. There's no partial charges. Again, basically growing in complexity. Alanine, next one has a methyl group. I'm not going to go through them all, but I'm just going to point out to remind you how some of the functional groups repeat themselves. Here we've got a growing chain, branching, so these are both 4 carbons, but the fact that they're branched differently makes them an isomer. That's why it's leucine and isoleucine. Methionine is going to be a critical amino acid when we start talking about how proteins are made. This is the very first amino acid that is coded for in any protein. Sometimes it's removed, sometimes it's not. We'll hear this again in a couple months. Phenylalanine is a fun amino acid because it's got a ring, fun chemically. It participates in a lot of hydrophobic interactions. It helps to stabilize the interior of a protein. Next, here we go. The polar side chain. So remember, the polar covalent bond is when there's a little bit of electronegativity difference. Again, we're going to have lots of oxygens in here, and we're going to have partial charges so that they are going to make hydrogen bonds. Even though there's a ring structure here, the very end has an OH, so he's going to participate in some hydrogen bonding. We're going to see in a second, hydrogen bonding is critical for holding protein shape together. Then there are the charged ones. Basically, these have organic acids at the end, or bases. So ones that are acidic, meaning that the pH of normal cell biology, they have lost their hydrogen, so they carry a negative charge. The basic ones have gained that positive charge, and these are all going to have a nitrogen that's grabbing it. Again, I'm not going to ask you to memorize the names. What they have here are the names. If you go on biochemistry, you'll probably have to memorize them and all the structures. We'll see sometimes the 3-letter abbreviation, and then sometimes they just go with a single letter abbreviation. We'll deal with this in a couple months when we do some worksheets with proteins and DNA. So let's start talking about shape. There's all different ways you can look at shape. We're going to do an activity where we'll play around with the shapes on the computer. This field has moved forward over the past 20-25 years tremendously. We've been able to crystallize proteins, figure out exactly where all those amino acid side chains are and how they're interacting with each other. You can do X-ray crystallography to obtain shape. You can do NMR, nuclear magnetic resonance, to figure out shape. And it helps us really understand how things work. Down here, my love of immunology makes me pick this example over any others. Mentioned antibodies previously. They're the proteins that are running around in your bloodstream, and they're there to help neutralize viruses, bacteria, and do some other features in your body to protect you. Just wanted to point out how exquisitely perfect this antibody molecule's shape matches the shape of a protein from a flu virus that it's there to neutralize. So it fits, it binds it because of some chemical attractions that are going to happen, and basically it gets stuck on it, and it helps to allow the antibody to do its job. And we're going to talk a lot about antibodies, probably not until March or April.